two trainees, um, and then a colleague from DPH. So just as a quick introduction to everyone, Leah Larissi is a PhD candidate in environmental health science at UGA, and Abby Wilson is a master's student in environmental health science at UGA. And they will be speaking first on some of their projects. And then we'll have Harrison Yu. Harrison's a senior bioinformatics specialist at Georgia Public Health Lab. He joined DPH in 2023 and works on wastewater sequencing analysis and bioinformatics infrastructure projects. So the way we'll structure this, Leah and Abby will go, we'll break for a couple of minutes if anybody has questions, and then we'll move into Harrison's talk, and then we'll have questions at the end. So I will let you get started, Leah. Thank you.
Yeah, thanks, Erin. Let me go ahead and share my screen. Share. Okay. All right, that should be good, but let me know if you can't see it.
Looks
Cool.
good.
All right. Hi, I'm Leah, and
Hi, I'm
that's
Abby.
Abby. And we're excited today to talk to you about some of our ongoing projects where we're applying wastewater-based epidemiology and doing some multi-target surveillance. So go ahead and get started. All right, so why wastewater? Well, clinical surveillance of infectious diseases is a necessary public health tool that lets us make informed decisions. But these data can be limited due to underreporting for a number of reasons. But wastewater surveillance offers us a way to overcome these limitations and gain community-wide insights on pathogen and antimicrobial resistance prevalence. And this is because we can obtain large amounts of data with minimal samples compared to clinical testing. And wastewater surveillance is independent of individual testing, so it's not biased by reporting requirements or healthcare seeking behavior. It's also pretty broad and non-invasive, meaning that it's not directly traceable to an individual or group of individuals. And so this is good for privacy reasons. And then also, this allows us to get basically real-time results. So avoiding some of those reporting delays that can be prevalent in clinical testing. Okay, so we're going to talk a little bit about our ongoing projects. So first, I'm going to talk to you about how I'm using wastewater as a tool to track local weather effects on enteric pathogen prevalence. And then afterwards, Abby is going to talk to you about how she uses wastewater to understand long-term trends and the emergence of antimicrobial resistance genes. So jumping into some background on our sample collection. So our wastewater samples were originally collected for SARS-CoV when we were doing like our SARS-CoV-2 monitoring. And so we collected samples from June 2020 through February 2024. And these were collected twice a week, so Mondays and Wednesdays, from 3 water reclamation facilities in Athens-Clarke County. And these samples are 24-hour composites, which means that they're basically subsampled at consistent intervals and then pooled over a 24-hour period prior to taking the final sample. So they're just representative over a 24-hour period rather than just a snapshot. Okay, and so how exactly do we go from raw wastewater to data? Well, first, we centrifuge our wastewater samples and we collect that concentrated solid pellet. And we then take that solid pellet, we mechanically shear it to release the nucleic acid and perform a total nucleic acid extraction using a commercially available kit. And so this allows us to pull out both RNA and DNA targets from the samples, which lets us detect a number of different targets. And so finally, we use quantitative PCR and specific gene assays to quantify each pathogen of interest. Okay. So now getting into more of the background of my project. So over half of all known infectious diseases, including enteric pathogens, are exacerbated by weather and climatic conditions. However, these findings are limited by the challenges that are present in traditional clinical surveillance. And even if those challenges or limitations did not exist, it would still be nearly impossible to capture clinical data at the same scale that we can capture weather and climatological data. And so, this is where wastewater comes in. Wastewater can act as like a location specific, highly resolved data source that we can match up to real time local environmental data and can fill gaps that are left by traditional surveillance. So we can answer more questions or more thoroughly. Okay, so getting into the specifics, so our basically our goal was to develop this proof of concept for examining effects of weather conditions on enteric pathogen prevalence in wastewater. And so we chose to focus on enterics because these are an especially underreported group of infectious diseases. And so uh, I basically pulled together this panel of 3 viruses and 2 bacteria. So, starting with norovirus, norovirus is causes like common stomach bug, and we chose to do the genogroup 2 just because it's more prevalent in the population than genogroup 1. We've also pulled enterovirus D68, which can cause respiratory illness, but in severe cases can lead to acute flaccid myelitis, which is a neurological condition. And then Coxsackie virus A6, which causes hand, foot, and mouth disease. Then we have some bacterial targets, Campylobacter and Salmonella, and these are commonly associated with food poisoning. And so that's those are our 5 pathogens that we looked for in wastewater. We also normalized those gene target concentrations to a human fecal marker in order to account for fluctuations in the population. And the fecal marker that we selected is PMMOV, and that stands for pepper mild mottle virus. Okay, so specifically for the study looking at enteric pathogens, basically, like during our original collection, our samples were partially processed for SARS-CoV-2 monitoring, but then the remaining wastewater was stored in a freezer for future use. And so we went back into our freezer stocks and we selected samples for analysis. We selected 155 samples from February 2021 to February 2024. And so basically we selected 1 sample per week from 1 water reclamation facility. And we chose this water reclamation facility that's circled in red. It has the second highest residential population in the county, but it receives influent from the 2 major hospitals. And so with that, we were hoping that we could get the data from the hospitals as well. So that was our thought process behind that. Okay, so getting into some of the results that I have, and all my results are preliminary. This is work that I have been conducting within like the past couple months. So, yeah, but I'm excited to share. So basically, the figure on the left is showing you the concentration of gene copies per liter of wastewater on a log base 10 scale, and that is just to make visualization easier. But we can see that over on the right, the fecal marker, PMMOV, was the most highly concentrated target in our samples. And that is a good thing because that means our extractions work well. It's wastewater, so if we have a lot of a fecal marker, then that indicates success. PMMOV, I guess I forgot to mention the figure on the right, but that is just showing detection frequency of all the targets throughout the entire time series. And so we can see that PMMOV was detected across 100% of samples, which is good. That means we can easily normalize our pathogen gene targets using this metric. So then looking at norovirus, that was also detected 100% and was the second most concentrated target, but the most concentrated pathogen that we detected. And so then moving to salmonella, we saw that detected in about 60% of samples. And then going further, we have the enterovirus, which we detected in about 25% of samples, and then Campylobacter and the Coxsackie virus were much less prevalent. Okay. All right, so now we're looking at the detection of our viral targets over the study period. And so these figures are showing you the PMMOV normalized concentrations. And so basically, that's just the number of pathogen gene copies per copy of PMMOV. So if you look at the y axis, that's why those numbers are so low, is because there's a lot of PMMOV, not a whole lot of pathogen copies in comparison. But um, anyways, so what really stuck out to me when looking at these figures was that there was lower prevalence in the norovirus and the enterovirus in 2021 compared to the rest of the time series. And so I wonder if this could be a result of social distancing measures that were put in place during the pandemic, or uh, yeah, I'm not sure, but it is interesting that we can see it in the wastewater. So then looking at our bacterial target detection throughout the study period, and these are also PMMOV normalized. Um, yeah, not a whole lot to say about this, but the key takeaway here is that we can detect a variety of pathogens in wastewater, even retroactively, like even in raw wastewater samples that have been stored back for years. So I think that's pretty cool. Okay, but now we're going to get into the assessing the influence of weather. Okay, so we have our pathogen prevalence data. We then downloaded local weather station data in Clark County, and we ran some additive models to assess the nonlinear relationship between weather variables and pathogen prevalence. And so we know that weather does have an effect on enteric pathogen prevalence because these concepts have been explored in clinical data, but we're now taking that and we're exploring it using wastewater data. So we basically tested different permutations of the weather data, starting with just the daily averages. But we also included daily minimum and maximum ambient temperature, as well as total daily rainfall. We also tested 1 week and 2 week lags, as well as a 7 day moving average, just to account for potential lag and cumulative effects. And so I won't spend a lot of time here, but these figures are just showing you the distribution of the different weather variables by month. And so this just gives us an idea of how these fluctuate throughout each season. And some of them, like temperature and solar radiation, do have like a very distinct seasonal trend that intuitively makes a lot of sense. It peaks in the middle of the year and is lower in the spring and winter months. So that's that. Okay, so now we get into something that's a little bit more interesting. And so these are some of the results from the additive models that I ran. And you'll have to take them with a grain of salt because these are very preliminary results and I'm still figuring out how to interpret them and have many next steps. But I'll orient you to the figures. They're basically what they're showing is norovirus prevalence in wastewater as a function of each weather variable. And so on the x-axis of each figure, you have your weather variable, and on the y-axis, you have the PMMOV normalized norovirus load. And the dashed lines are representing the 95% confidence intervals that surround the model predictions. And so when we look at the figure on the left, we see that norovirus tends to be more prevalent when minimum daily temperature is low, although there are some slight variations that occur as temperature rises. And this makes sense, given that norovirus cases tend to be more prevalent in the winter months, although they do tend to pop up in the summer as well. And so, although the confidence intervals are a bit wider on that graph on the right, which is showing you solar radiation, there also seems that like this variable is also somewhat predictive of norovirus prevalence. And so what's interesting about this is it's a 1 week lag. And so we didn't see this relationship when we assessed average daily solar radiation with no lag. But we did see it with the 1 week lag. And so that is indicating that there is some sort of lag effect going on. Okay. And just to briefly conclude, these results indicate to me that wastewater is a promising tool for exploring impacts of weather conditions on infectious diseases, specifically enteric diseases. And I have some next steps that I'm working towards. So, my first is to get some clinical data so that I can compare the trends in clinical data to those observed in the wastewater. And then I'd also like to build some multivariate models to get a better assessment on the combined effects of these weather variables. And yeah, that is what I have done so far. And so with that, I'm going to turn it over to Abby and she'll talk to you about her research.
All right. Hello. My research project focused mostly on using wastewater to um, analyze both long-term trends as well as the emergence of AMR. So I won't focus too much on the background, as I'm sure everyone understands mostly the basics of what AMR is, or antimicrobial resistance. But essentially, with the global overuse and misuse of antimicrobials in agriculture as well as in veterinary and human healthcare settings, we've seen an increase of the evolution of AMR. This figure on the right is showing from the CDC AR report update from 2021-2022. And we can see there are some urgent emerging threats, including carbapenem resistance. So understanding and having surveillance for these emerging threats, as well as what resistance is occurring in the population, is important for antibiotic stewardship programs as well as guiding policy. So currently there is clinical sampling that is done through governments as well as alongside the CDC. However, collecting these clinical samples is important and understanding what's happening on an individual basis, but we do get a limited scope overall, and we're missing a large chunk of data, such as those that aren't seeking treatment in healthcare settings, but are still harboring these resistance genes in the gut. So that's where wastewater-based epidemiology can come in for understanding AMR. So the scale of analysis that we can achieve with wastewater-based epidemiology is nearly impossible with clinical sampling, as we can gain a community-wide AMR profile of what sort of AMR genes are fluctuating and circulating within the population. And we can also hope to receive some information on early detection of emerging threats, including those urgent, critical AR threats before they pop up in clinical samples. So for my specific methods, I used a similar pipeline. I concentrated the wastewater influent samples that were frozen back. I extracted the total nucleic acid and then ran duplex qPCR to relatively quantify 10 total AMR genes from a total time series of June 2021 through February 2024. And I was focused on a different wastewater treatment facility. As opposed to Leah's research, I was using the North Oconee wastewater treatment facility. And the importance of this was just I was trying to capture student population dynamics, as this catchment facility receives influence from campus area as well as downtown. It does receive a little bit of industry. However, I was hoping to mostly capture some student dynamics. And then these were, again, retrospective samples. I did one sample per week, ran in triplicate in these duplex qPCR assays. And then at the very end, I normalized my AMR gene concentrations to another human fecal marker, HF183, to hopefully understand how much AMR is there in our waste relative to human population shedding. And so these were the AMR genes that I chose to focus on. So the six that are non-bolded are six more commonly found genes within this area. So they were found already in previous studies at very high concentrations in the Athens-Clarke County watershed before. So I was hoping, or I expected to find them at higher concentrations to hopefully use them to gain some seasonality insights as to how these genes have been fluctuating over time, over a long period of time. And then the red genes that are bolded are for rare emerging targets that have been alerted to us as emerging threats and urgent threats by both the CDC as well as Georgia DPH. Specifically, the beta-lactamase NDMs and the OXA48 confer resistance to carbapenems, which are last resort antibiotics that are used to treat very severe infections. And the MCR1 also confers resistance to another last resort antibiotics. So I chose to focus on six more commonly found targets to hopefully gain some seasonality aspects. month by month or throughout the year. And then the 4 red targets I chose to focus on to see if they're emerging in our area as well. So the first figure I want to discuss is detection frequency overall. So this was the positive rate for all of my targets for all of my samples. I had a total of 126 wastewater influent samples. And as you can see, those 6 that are that I expected to be very common in our wastewater, they're very common. 5 out of the 6 commonly found targets in the watershed were found in 100% of my samples. And then the beta-lactamase CTX-M1, another common one, was found again, fairly common in 97% of my samples. And then the 4 rare targets that are on the right side of the graph, I was still able to detect them in my samples at some capacity. But the one that is interesting is this beta-lactamase OXA48. That confers resistance to a last resort antibiotic, carbapenem, and I was able to detect that in 66% of my samples. So a little bit higher than some of those other rare targets, the NDMs and MCR1. So it could indicate that it's starting to pop up a little bit more frequently in our area. And this figure I'm showing now is the average normalized concentrations for each of my targets monthly across the entire time series. And these are normalized to HF183, that population marker. And then I logged this, this data as well. So the months that are blank, so where there's no line or data point, including the rare targets that are down below, this means that all the samples in those months were non-detects. So I took them out for now. But we can see that overall, the more commonly found targets, those 6 more commonly found targets, they do have some potential seasonal variation, which I'll get into in the next bit. But overall, we do see they're fairly consistent in our population over the entire time series. They are pretty consistent up top at higher levels. Possibly indicating they're just circulating pretty frequently. And then those rare targets are down below. They have a little bit more sporadic episodic nature. But interestingly, that beta-lactamase OXA48, it does have a little bit of sporadic episodic nature in the middle/beginning of the time series, but it starts to level out towards the middle slash end, so 2022, 2023, possibly indicating that it's starting to circulate more consistently in the population, just at a lot lower levels. And those rare genes have more introduction events where it's a little bit more sporadic and episodic in nature. So as I was saying, those 6 more commonly found targets, I wanted to hopefully assess some seasonality with those. So just as an example, I pulled the class A beta lactamases. This figure is showing you monthly averages across all the years in a box plot format for each month. And then this is also normalized to HF183 as our population marker. And we can kind of start to gather a little bit of seasonality data from these class A beta lactamases. We have beta lactamase KPC and SHV on the left-hand side. Those show a slight peak in August, maybe late summer, early fall months, whereas the beta lactamase CTX-M1 does show a little bit of a peak in August potentially, but it does show a little bit more of a peak in late winter, early spring months. So these are just a little bit preliminary for now. I do want to do some more statistical testing to see if there's statistical significance in that monthly variation, but just kind of starting to look at some potential reasonings as to why we're seeing some peaks in certain months. Something that would be interesting to look at is if these beta-lactamase KPC and SHV peaking in that August time period could potentially be students coming back from summer, causing that seasonality. So just to conclude, I was able to find all 10 AMR genes that I was looking for in the wastewater samples at some capacity, with those 6 more commonly found targets, 5 of them being found in 100% of my samples, and beta-lactamase CTX-M1 being found in 97%. So they're definitely consistently there in the population. The rare targets I've found more episodic, potentially emerging over time, with specifically that beta-lactamase OXA48 possibly stabilizing and becoming a little bit more consistent, but at lower levels. And overall, wastewater-based EPI can be great to fill in some gaps that clinical sampling possibly leaves behind. So in combination, we can hopefully create a full AMR picture of what's happening at the community level and in a population level. And some next steps that I would like to focus on as well. Again, I would like to test for statistical significance in that monthly variation to see if there is some actual significance there in the variation. And then I would also like to compare to clinical cases to see if there's some sort of comparison that can be made to some seasonality and clinical AMR reports. And then I would like to further some of my time series analysis. So thank you so much for coming to our talk. Here is our contact information.
Thanks.
Please email if you have any questions, or we can take any questions.
Thank you both. So I think we can like hold just a couple of minutes for questions if anybody has any before we move to Harrison's talk. You're also welcome to put things in the chat if you want. And then of course at the end of this we can have questions for all of the speakers.
I'm gonna go ahead and
Okay, so I think we will go ahead and move to Harrison. So thank you, Lee and Abby. That was great. Harrison, if you're ready, we're happy to see you start.
Sure. Can you guys see my screen?
Now we can.
Okay. Yes. Hello, everyone. My name is Harrison Yu. I'm the senior bioinformatics specialist here at Georgia Public Health Lab. I'll be talking about our method of detecting SARS-CoV-2 variants in wastewater samples. So the lab receives wastewater samples from 17 collection sites across 16 counties in the state of Georgia. We perform extraction using the Kingfisher Apex platform. Library prep is done using the Illumina COVID-seq assay with Arctic V5.3.2 primers. And we do paired end sequencing on the Illumina NextSeq 1000 and 2000 series of sequencers. So I'm going to be getting into a tutorial on how to use a bioinformatics tool called Freya. This is a variant caller that is designed to look at mixed samples that are normally associated with wastewater. So the prerequisites for using this tool, most of these tools that you see listed here are found in any typical variant calling bioinformatics pipeline. You typically you start out with a QC step to generate QC metrics. You'll perform some kind of adapter trimming, scrub human reads, and align to a reference genome. We then filter and trim primers from the alignment file in BAM format, and this file is what Freya will look at. So for installing this tool for Hypergator users, you guys are able to use Aptainer or Singularity on the cluster. And for all other users, I'd recommend using Docker, and Conda are also available options. So before using the tool, we'll need to download the most recent lineage data. There are two ways to do this. You can run the command Freya update, and that will go ahead and fetch the latest barcode information, or you could download the two files that you see there from GitHub manually. So the first step is to perform variant calling using the reference genome FASTA and trimmed and filtered BAM files as input. And to generate the lineage abundances using the variant depth, usher barcodes, and curated lineage files from earlier, we'll use this following command that you see here. So the result of this tool, the demix tool will produce the following file that you see over here on the right, and this will contain the lineage abundance information for each sample. So the lineages array contains the sample's identified lineages, and the abundances row contains the corresponding abundances for each lineage that was called. I also recommend combining files from multiple samples using this command right here. So from this result, what we can do is we could plot the lineage abundances for each sample, and we can group them by the collection site that each sample came from and sort them by date of collection. So what this will tell you is if any new and emerging variants are introduced to a particular site or a region. So Freya can be used to identify emerging COVID-19 variants in wastewater samples, detecting them in communities before clinical cases are reported. So public health officials can get an early warning of new variants circulating within a population and take appropriate actions. In order to do this effectively, a routine sampling schedule, like once or twice a week, is crucial for establishing a reliable baseline, which makes it possible to detect introduction of a new variant. For efficient processing for multiple samples, I recommend using a Nextflow pipeline combined with a cluster or a cloud computing system. So what we use now is CDC's Aquascope pipeline with a few modifications, but this is one example of a custom pipeline that people can use. So we do have future plans going into 2026 for sequencing. We are considering moving towards targeted metagenomic sequencing. The plus side to these things are we can target multiple viruses using a probe capture approach. This offers improved efficiency over running separate tests for each virus. It generates more comprehensive results, so it allows us to identify novel viruses. And we are also considering long-read sequencing on the ONT GridION. And this is currently in the works. So I think this is the last slide. Yeah. So I'm ready to take questions.